And share my screen. Okay, so um, here's the list of topics that are going to be on the exam according to Jake, and then I kind of based my review off of what's going to be on the test. So the first topic is control systems. And for control systems, you want to be prepared for first knowing what positive versus negative feedback is and being able to recognize it if you're given a diagram. So here's an example. Is this diagram showing positive or negative feedback? So first of all, what do those two things mean? So both of them refer to when a change happens and then some kind of response occurs in the body. If a change happens and the response is to keep going in that same direction of the initial change, that's positive. If a change happens and the response is to work against that change, that would be negative feedback. So here we have a diagram that says, baby's head presses against the cervix, causing it to stretch. In response, the stretch on the cervix causes action potentials to be sent from the cervix to the pituitary gland. And then the pituitary gland in response releases oxytocin. And the uterus has receptors for oxytocin, so it responds by initiating uterine contractions. Uterine contractions cause the baby's head to press against the cervix, causing it to stretch, and the whole cycle starts over again. So you should recognize this as being a positive feedback loop because the initial change, which is the baby's head pressing against the cervix, initiates a response that causes more of that change. The baby's head continues to press against the cervix. And this is how contractions in the uterus happens and how babies come out. So this is positive feedback. And then next thing you should be prepared to do on the exam is if Jake changes something about this diagram, can you predict how other parts of the diagram will change in response to the change he made? So my example was, if the stretch receptors on the cervix are blocked, how would oxytocin levels change? And also how would uterine contractions change? So if you block the stretch receptors on the cervix, then there's not going to be any action potentials in response to the baby's head pressing against the cervix. If there's no action potentials, then there's no signal to the pituitary gland, and so oxytocin is not going to be released. So oxytocin levels should go down. How would uterine contractions change? If there's no oxytocin release and no oxytocin stimulation of the uterus, then there won't be uterine contractions. So as a result, you would get less of uterine contractions, oxytocin release, and overall less positive feedback. Another example question would be if oxytocin receptors on the uterus were blocked, how would the amount of stimulation of the pituitary gland change? So if you block the oxytocin receptors, which you should recognize as being on the uterus, then you wouldn't get contractions, you would get less pressing against the cervix, less action potentials, and then therefore less stimulation of the pituitary gland. So again, less overall positive feedback. So that's an example of what you should be prepared for. Given a diagram, can you tell me if it's positive or negative? And then can you answer questions about how different parts of it will change if I block certain parts of the pathway? The other thing that you need to know is that feedback pathways that involve hormones, in this case oxytocin, are specific, meaning oxytocin doesn't just signal to all parts of the body, but it specifically is signaling to the uterus. And you need to know, how does this specificity arise? This came up in Jake's review, as he said, like, how do organs know to respond? How do the correct organs know to respond to certain signals? And the answer is just that if organs have the correct receptors for those specific hormones, then they will respond. It's not that the oxytocin hormone knows where it's going, right? It doesn't only travel to the uterus. It travels all over your body, but it's only going to cause a response in the tissues that have receptors for that hormone. So that's a point to note. Okay, so here's another example. And this is one hopefully you're all very familiar with. So you should decide, is this diagram showing positive or negative feedback? So here we have hypothalamus releasing TRH, which stimulates the pituitary. Why? Because the pituitary has receptors for it, which causes TSH release, which then stimulates the thyroid to cause T4 and T3 release, which causes increased metabolism. And we can also see that T4 and T3 goes back and inhibits the pituitary and the hypothalamus. And also that TSH inhibits the hypothalamus. So when you see like a line and then a, I don't know, horizontalish line, that indicates inhibition. Yeah, I see there's a raised hand.
We don't have to remember this diagram, right? He would probably provide it. We just have to know how it works.
Yes, in fact, both diagrams that I'm showing here are likely not going to be the ones you see on the test, but the same exact idea. You're going to be given diagrams you've never seen before, we've never talked about.
Okay,
but
that makes sense.
follow the same patterns. Yeah, so this one you should recognize as negative feedback because there's some change, which is to increase metabolism to produce T4 and T3. And the response of this system is to decrease that change, to inhibit the process that causes increased metabolism and increased T3 and T4. So your big giveaway for whether a diagram is negative feedback is, does the thing that results in the end go back and say, stop doing that? Or does it go back and say, do that more, like we saw with the uterine contractions? So here it's saying stop, so it's negative feedback. And then again, be prepared for something in the diagram is going to change. Maybe we block the TRH receptors. Then you should be prepared to tell me how other parts of the diagram are going to change. So if we block TRH, how will that affect TSH? Well, with less TRH, then that's going to stimulate pituitary less. So that will cause TSH levels to decrease. And also, how would T3 or T4 levels change? T3 and T4 levels would also decrease. So you could imagine maybe a change in the opposite direction. Maybe there's increased expression of TRH receptors. Then you would expect more release of TSH and more T4 and T3 and more negative feedback. Another example, if TSH receptors are blocked, how would TRH and T3, T4 levels change? So if we block the TSH receptors, where are TSH receptors? Well, they're going to be on the thyroid because that's where it's signaling to. Again, not because TSH knows to go to the thyroid, but just because the thyroid has receptors for TSH. So if there's less TSH receptors on the thyroid, then that's going to cause T4 and T3 levels to go down, and that will cause less inhibition of the hypothalamus and pituitary. And if there's less inhibition of the hypothalamus, then there should be more TRH. So kind of working through that thought process is what you'll be expected to do on the exam. Any questions about control systems? Cool. Okay, then next we have scaling. And he specifically wants you to know about log versus linear axes and how dimensions change with body size. So scaling law in biology is when you look at one variable, it's proportional to another variable to the nth power. And the degree of the proportionality depends on whether it's a one dimension thing, 2-dimension thing, or 3-dimension thing. So that's kind of what we're talking about with scaling laws. So first, let's talk about log versus linear axes. First, you need to know what is a log scale axis versus a linear scale axis, and can you identify each if you're given some graphs as examples. So a linear scale axis is one that increases by addition. So as you go from one to the next, it increases by some number. And when you go to the next number, it should increase by that exact same number. So for example, here I have tolerance range. You can see that going from 25 to 30 has increased by 5. The next number also increases by 5. The next number also increases by 5. That is a linear axis. It's increasing additively by the same amount. A log scale axis is one that increases by some multiple. So over here, you can look at energy spent per day. It's going from 10 to 100. So if we were trying to see whether it's linear, we'd say, okay, it's increased by plus 90. If it was linear, the next one should also increase by plus 90. But you can see that it does not. It is not 190 here. Because it's increasing not by addition, but by multiplication, this is a log scale axis. So for every axis here, you should be able to identify whether it's log or linear. So here, let's try to identify this one. So it says energy spent per day. We have 250, and then we go to 500. That's increased by 250. We go from 500 to 750. That's also increased by 250. 750 to 1000. That's also increased by 250. We know that that is a linear axis. Here on the bottom, we've gone from 10 to 100, again, plus 90. If we did plus 90 again, we should have 190 here. We don't. We know this is a log axis. So this one is log. This one's also log. If we go down to latitude here, 10 plus 10 is 20. 20 plus 10 is 30. 30 plus 10 is 40. This is also a linear axis. So what I just did, you will be expected to do on the test. Identify which is log and which is linear. You should also know, why does it make more sense to use a log versus linear axes? The advantage of using a log axis is for spreading apart data when you have data that occurs over large magnitudes. So you can see in this first graph, a bunch of data points are all bunched together because we are using a linear scale. So it makes it look like this data isn't all that different because we had to also graph these other points that are really far away. Because these were far away, they made these get all bunched together. And now the human brain, when it looks at these data points, it thinks that these aren't very different. So a log scale, this is the same data, but with a log scale, allows it to spread data that occurs over many magnitudes apart. So you can see differences better. And then the third thing you should know is, given a trend line, kind of like we have here, can you determine which data points seem to follow scaling laws and which don't. And so basically what that means is, can you determine based on the linear line whether the data is following that linear line or not? And what you want to look at is not only how far the points are from the line, but how far they are and how far generally all of the points are. So what I'm saying is, although these points are pretty far from our linear trend line, we can see that all of the data is pretty far from the line. So I wouldn't necessarily call these data points outliers, because on the whole, all of them are pretty far apart. If you look at this graph here, if I were to draw a line through this data, and then I asked whether like these 2 points were outliers, I would say, yeah, those are more of outliers than these are, because the majority of the points are going to follow that linear line. So deciding whether points are kind of unusual or not following scaling laws is not just how far away they are from the line, but also how far away they are in comparison to all of the data that's being shown. Okay, questions about log scales and graphs?
Sorry, I think you already said this, but can you define again what is the log scale axis versus linear scale axis, like the definitions?
Yeah, so a linear scale axis changes additively by the same amount, whereas a log scale changes by some multiplication of some number. I think the real word, and I avoid saying it because I can't say it, but it changes multiplicatively. But I can't say it. It changes by a multiple. So like, for example, if you look at this graph here, each number, if you times it by 10, that gives you the next number. But you can't add some number and get the next one each time.
Yeah, that makes sense. Thank you.
You're welcome. Okay, so the next thing he wants you to know about scaling is, can you predict how dimensions change with body size? And again, this is just those scaling laws. And basically what you'll need to know is this chart here. So you should know that one-dimensional things will change by some factor n, two-dimensional will change by n squared, and three-dimensional by n cubed. So this is just something you'll have to know. But the actual problems you'll be expected to solve are on the more straightforward side, I think. So if you were able to do the worksheet with the salmon, you'll be pretty prepared for the test. So here's just another example. It says a baby kiwi weighs 0.2 pounds and is 6 inches tall, and an adult kiwi is 1.5 feet tall. By what factor has the kiwi increased in height by adulthood? So by what factor has the kiwi increased in height? So height is one-dimensional, so that factor is just going to be straight up in. So if you compare how the height has changed, it was originally, yeah, there's a question?
I was wondering, are calculators allowed on the exam? I don't think it was clear.
I'm gonna say yes, historically. And I think like on Canvas, it always says like, bring a calculator, bring a calculator.
Okay.
Yeah, I'm pretty sure yes.
Thank you.
You're welcome. Okay, so by what factor has the kiwi increased in height? It's one-dimensional, we're looking for in. how is the height changed? 6 inches to 1.5 feet. So first you have to convert it into the same unit. So convert 1.5 feet into inches. So that would be 18 inches. And so 6 times what equals 18 inches? Or just divide 18 inches by 6. And you know that the factor has increased by 3, right? N equals 3 because 6 inches times 3 equals 18 inches. Then you should be prepared from there to also be asked about 2-dimensional and 3-dimensional things. So how much should an adult kiwi weigh? That's mass. So that's a 3-dimensional value. We already know what N is. It's 3. So an adult kiwi should weigh whatever the baby kiwi weighs times 3 to the third. So what you would do is 0.2 times 3 times 3 times 3, and you would get 5.4 pounds. And that would be your answer. And then
Would it be a good idea to round up the factor first, or just keep it as it is and then round afterwards? Because most of the time you get like some kind of decimal point and not like a clean number. So, and if you round it or keep it, you can get different values depending on which one you use. So I was wondering if that would be something to consider.
yeah, so I would try to round not till the end. And I'm pretty sure that for the test, rounding won't be an issue.
Okay, thank you.
Quick question.
Yeah,
You said it's 0.2 times 3 cubed, right?
yeah.
Okay, thank
So you should get 5.4. And then it says, by how much should a kiwi's surface area increase? So then you have to think, okay, surface area is 2-dimensional, so it should increase by 3 squared, or 3 times 3. So it should increase by 9 times. So that kind of basic dimension changing is what you'll be expected to do. Last thing with scaling, can you identify scaling law assumptions or when scaling laws break down? So it's important that you know that these scaling laws are really useful and they work a lot of the time, but they're not infallible, right? You can't just say, well, if the scaling law says the surface area is going to increase by 9 times, then it is for sure that the kiwi surface area is going to be 9 times bigger. That's not always the case. So what kind of assumptions should you be aware of? So the big one to be aware of is body composition. So when you go from a baby to an adult animal, or if you're comparing 2 different animals, one that's small and one that's big, a huge assumption that the scaling law does is that the body composition is the same. They have the same amount of fat and muscle. So because of that, your actual values for, like, say, mass might be off. So yeah, just be aware that there are assumptions and they can break down, such as in the example of body composition. And that is it for scaling. Any questions on scaling?
Hi, could you repeat the math that you done for the the kiwi weight one more time?
Yeah. So we got from the height what in was, because height is one-dimensional, and it was increasing by 3 times, right? If you do 6 times 3, you get 18 inches. So then when we're looking at the mass, we know mass is just in to the third. Since we already established in was 3, we knew that the mass had to increase by 3 to the third. So what you would do is you would take whatever the small mass is of the baby kiwi, which is 0.2 pounds, and you would multiply it by 3 to the third. Or that's the same as 0.2 times 3 times 3 times 3. And then that gets you 5.4.
3 to the third. Okay, got it. Thank you.
You're welcome.
I have a quick question.
Yeah.
Did you see the exam?
I have taken a look at the exam, yes.
I was wondering, did you make these slides after you saw the exam?
Yes, I tried to tailor them to the exam, yes.
Okay, thank you.
All right, any other questions on scaling?
One more. How do you tell that like the difference in question 2 for the kiwi question from question 2 to question 3, where it says how much does an adult kiwi weigh? I calculated 5.4, but for the next one, for the surface area, I also calculated the value instead of like by how much it changes. How on a test, like if they don't write by how much, like how would you differentiate between the values? 'Cause both questions seem very similar.
Yeah, so if I was asking you what is the surface area, I would have to say, tell me the surface area, not by how much is it going to change. Yeah, you just have to be careful. And that's something you could raise your hand and ask me during the test. You could ask, is it asking me for how much or an exact number? And that's something I could tell you.
Wouldn't you also have to be given the surface area of the baby kiwi in order to calculate the adult kiwi surface area?
Yeah, exactly. So I can say that it's going to go up by 9 times, but because I don't know what the baby's was, I wouldn't even be able to calculate a bigger surface area either.
So to
I
calculate
think you
the
can. Like if you use, because you're given mass, and then you can use the relationship between surface area and mass to determine the surface area for the adult kiwi. So it'd be like the surface area of the baby.
Oh, that's true. You could do that. That would be tricky.
Yeah.
But yeah,
You'd have to like put it to the power of 2/3, I think, or 3/2.
yeah, but you could do a ratio. So you could do surface area to volume. So it'd be N2 over N3 equals X over 5.4 and create a ratio to do that. So yeah, I guess technically you could do that.
wait, can we go over that kind of example?
Sure. So like just how would you calculate the surface area here?
Yeah.
Yeah, so I guess what I'm saying is we know surface area to volume ratio is N squared over N cubed, right? We know that's how it should change, right?
Yes.
And then we don't know what the surface area is, so that's X, but we do know what the volume or the mass is, right? So we could put 5.4. We also know what N is. So then you would just solve for X. So you could solve for what this number is and then multiply it by 5.4 and that would give you X. So that would be pretty tricky, but yeah, I guess technically you could do it.
So we would find X and then again put it in the ratio, right? To get the
Well, if you're asked for just the surface area, then that would just be X. Because it's like
Oh,
surface
I see.
area
Okay.
to mass
Yeah.
ratio.
I see. Okay, thank you.
You're welcome. Yeah, so that's it for scaling. Then temperature stuff. So big picture, what do you need to know about temperature stuff? First, you need to know the difference between endotherms and ectotherms. So the big difference is that endotherms regulate their temperature internally using their metabolism. Ectotherms do not internally regulate their temperature. Their body temperature just matches whatever the external temperature is. So ectotherms tend to not live in extreme temperatures because they are going to be whatever temperature it is outside. So then the next two things to know are, how do these two types maintain ideal internal temperatures that will allow them to function if the external temperature varies, right? So the enzymes in our body only work within a certain temperature range. So how do both types keep themselves within that range? So let's think about endotherms. So endotherms, let's say they're in a extremely cold environment. What they're going to do, or one of the things that they'll do, is actually have like a physical response where they will shiver to increase their body temperature. They will also constrict their blood vessels. So that causes them to increase heat inside. And they can also change their metabolic rate. So they will increase their metabolic rate, and that will generate heat for their body. And same thing, just in the opposite direction, if they're in hot environment, right? They will decrease their metabolism, they will dilate their blood vessels, and they will sweat. Yeah. Question.
So you said endotherms like regulate their temperature internally using their metabolism, right? But even for ectotherm, you're saying like they would increase their metabolic rate. So how would that be different?
No. I'm not saying that for ectotherms.
Oh, so shivering and constricting blood vessel, all of that, was that for endotherm?
Yeah.
Okay.
Everything I just said only applied to endotherms.
And how does like constricting blood vessel generate heat?
Constricting blood vessels just decrease heat loss. So when you have wider diameter blood vessels, more heat can flux away from the blood vessels.
Do we have to know how?
No.
Okay. Thank you.
You're welcome. How do ectotherms maintain ideal internal temperatures to function if the external temperature varies? So they're going to do more behavioral things. They can't change their metabolic rate in response to changes in external temperatures. They're going to have to move to different areas. So if it's, if there's sun shining, they might literally just move to a more shady area. But the big distinction is endotherms can change their metabolic rate to try to regulate their internal temperature. Ectotherms don't have that ability. Questions on temperature?
Is this the same like concept as tolerance, range, and performance embedded in that graph, or do we need to know that graph for this part or no? The ectotherm versus endotherm. Like the endotherms have a smaller or have a larger tolerance range and a like a wider performance breadth, things like that. Do we need to know or no?
I mean that does sort of relate to the concept that ectotherms, they're going to be whatever temperature it is outside. Whereas endotherms, that's not necessarily true. They can do things through metabolic processes to change their temperature so that it's not necessarily as cold or as hot as it is outside. So that's kind of what you need to know. Yes, Hannah.
So ectotherms can't change their metabolic rate at all.
No, they cannot change it in response to temperatures. They can change it, like if they're running versus sleeping, then their metabolic rate is going to change, but not as a way to regulate their temperature.
Okay, thank you.
Yeah, question?
So the like animals who like hibernate, are those usually ectotherms?
No, they're usually endotherms.
And can you explain why?
Yeah, that's just another behavioral response to trying to conserve heat during really cold times of the year. So it's not necessarily one of the responses that have to do with changing metabolic rate. It's just a behavioral response of some endotherms. It's more specific rather
I
than.
see, but like they regulate their temperature using metabolism, right? So like when they hibernate, they wouldn't like eat anything. So like how are they maintaining? Just like using up their fat, and like once it's used up, they just come back.
Yeah, using up their fat and decreasing their activity significantly.
Okay, thank you.
You're welcome. Cool. So then the last big topic is sensation. Okay, so for a sensation, we're going to focus on vision. So you need to know what electromagnetic radiation is and black body radiation, not because you're going to be asked to define those on the test, but it's going to be helpful to understand what these things are in order to know what's going on with vision. So electromagnetic radiation is all of the wavelengths of light. So you need to appreciate that there are many wavelengths of electromagnetic radiation beyond what is just visible to humans. And the reason why we can see that specific range of electromagnetic radiation is just because of the photoreceptors that we had. If we had different photoreceptors with other properties, we would be able to see maybe other wavelengths of electromagnetic radiation. And in fact, there are other animals that can see a different range of wavelengths, right? So like bees can see in the UV range, and there are snakes that can see in the infrared range. Black body radiation, what you need to know about that is that objects will radiate light depending on their temperature. So the sun is radiating light depending on its temperature, but also animal bodies will radiate light depending on their temperature. So what's the importance of that? Our bodies, when they are radiating light, we can't see that light. Why? Because that light is in the infrared range. We can see the light coming from the sun because it is within our visible range. But maybe if we had some mutation to our photoreceptors, it would be possible to see the light that radiates out of bodies in the infrared range. Yeah, so that's kind of the main concept with black body radiation, is that objects radiate light depending on their temperature. And also appreciate that what humans see generally is not the light that is radiating from most objects. Most of what we see is reflected light. So when you look at an object, it's reflecting light from some other light source, usually the sun. What are photoreceptors, and what specific portions of the spectrum can human photoreceptors sense, and how can you tell? So photoreceptors, those are the proteins and the cells in our eyes that have those proteins that can absorb specific wavelengths of light. And here you don't have to know like the exact wavelengths of light, like the fact that it's from like 400 to 700 nanometers. What you need to know is the only range of wavelengths that we can see is the wavelengths that our cones can actually absorb, right? The reason why we can see those wavelengths is because we have pigments that respond to those wavelengths. If our photoreceptors do not create a response to the wavelength, then we're not going to see it, right? So that's kind of the answer to how can you tell. With our cones, we also have the extra ability of color vision, because our eyes with these different types of cones can now compare the relative activity of these different photoreceptors and then say, what is the ratio of each of the wavelengths that we're looking at, right? So the reason why we don't have color vision at night is because the only photoreceptor online at night is the rods. And I haven't showed the rod's absorption spectrum here, but the important point is there's only one absorption spectrum for rods. And if it's just one, then there's nothing for the rods to compare their activity to. In order to see color, you need to have more than one photoreceptor, and your brain needs to be able to compare those two. Yeah.
So are you saying like at night cones are turned off and only rods can see?
Yes, exactly. Yeah.
But even during day, right? Like cones like absorb like different lights and like it's able to like interpret colors. So like how come rod doesn't do that?
So rods also, once the light, ambient light reaches a certain level, they are no longer, they're no longer able to sense changes in light. So they're very sensitive and they kind of max out their firing very quickly. So they're able to sense small little changes in light, which is great for the dark. But once you get to even just like evening light, they're firing at their max value. So as you go from evening light to daylight, they're not telling your brain anything anymore. They're saying the same thing at evening light as they are at daylight. And if you don't have a change, then it's basically not telling the brain anything.
So it's sensitive to a specific amount of light.
Yeah. And if we had like two rods that had two different absorption spectra, then we could presumably get color vision at night. But we don't. We just have rods and they all have the same absorption spectrum. So they can't
So
compare.
like, but I see, but cones does have different absorption spectrum.
Exactly.
Okay. Okay, that makes a lot.
I
Yeah.
have a question about the cones and rods as well.
Yeah.
So I'm getting conflicting answers from different like TAs and professor. So you said you need a combination of rod or cones to see color. But like on the slides it says color is like the evolution of the olfactory sensor, and then for the olfactory sensor, it had the example like if you have three olfactory sensors, then you can see seven, like you can have seven smells because one for each, and then the combination of four, so that was seven. And then the professor told me that you can also see with like a single rod, but it's not going to be very clear. Like if you just have the blue rod, or sorry, the blue cone, then you can see all the, like you can see cyan, but it would be really hard to tell that it's cyan without the red. Like it would be hazy or something, is what he mentioned. I just wanted to like, um, but then the TAs are all saying that you need multiple like cones to see color. So it's conflicting answers. I was just wondering which one's correct.
Yeah. I mean, as someone who works in a color vision lab, I can tell you if you have one cone, you will not have color vision. You need to have multiple. That's just how the brain is wired. It literally, we know that it compares cone inputs, and if it only has one, you see in black and white. It's rare, but that's true. You don't see color if you have one cone.
Awesome. Thank
I have a question about rods. So I read in a textbook that rods are like dark sensing cells. So basically, the way it senses light is by sending less action potentials to the brain, and the brain interprets that as sensing light. Is that right?
Yeah. So
like it's
go ahead.
yeah.
Yeah, so basically what all of these do, whether it's rods or cones, is they get a photon of light, and in response to that photon of light, they actually close sodium channels. So they hyperpolarize to light. And the more photons of light there are, the more they hyperpolarize. But you can imagine rods, if one photon comes in, they close like 300 channels. And then if two photons come in, they close like 1,000 channels. And then if they only have 1,000 channels, they can't sense any more light past that point. So they're very sensitive, but they go offline pretty fast. Whereas cones, they might not even really, well, that's not true. They barely respond to one photon of light. They might close like 2 channels. There's barely response. But that allows them to have changes in their responses over many photons of light in a way that rods can't. But this is not on the exam.
Okay, thank you.
You're welcome. Yeah, Anthony?
I was just wondering how color blindness works. Like, is it just like a lack of a certain cone, or is it like a cone being cut short or something?
Yeah, exactly. So color blindness results in when someone has a mutation and they're missing one of their cones. It's very rare to be missing multiple, but the red and the green cone, or the L and the M cones, they're both on your X chromosome. So it often affects males more often because males only have one X chromosome. So if they're missing one of their cones on the X chromosome, they're going to be colorblind. Whereas females, if they have on one X chromosome, they're missing one, but the other one they have it, then they're good. So that's why colorblindness is often seen in males. And yes, if you're interested in this stuff, there is a lab you can join. Okay. Question, yes.
So missing, I'm just confirming what you said. So missing either L, M, or S cone, like even just one of them would lead to color blindness.
Yes.
But that would be just color blindness of like specific color, right? The person would still be able to see some colors.
Yeah, so you would have to have only one cone to see in black and white. But most people who are colorblind, they're missing their M or their L. And so what they normally see is blue and yellow.
I see.
So you can imagine they have their S cone and they see all S cone responses blue, and then they have one of these two, and that's just interpreted as yellow. And they can't see the differences between like green and red because they don't have two different cones to compare in that area.
I see. So S is for blue, and then L is for green, and M is for red. Okay.
Yes, and in a simplified way, that's fine for this class. But technically it's, yeah, short, middle, and long wavelengths. Because if you look at like the red cone, it's technically firing most to like orangish light. So scientists, they don't like you to say it's red, but it works for this class. Oops. Okay.
Well, I had one more question about this.
Yeah.
So what did you say if for 3, what color would you see if you stimulated your M and L about equally?
Yeah. So first,
is that about green or like greenish yellow?
Yeah, let me go through both of these questions because we kind of drilled, but these are things for the test. So can you predict what colors you wouldn't be able to see if you lost L, M, or S cones? In a very simplified way, if you lost your S cones, you would be missing whatever wavelengths of light are unique to the S cones, which are like bluish colors. If you were missing your L cones, you wouldn't be able to see kind of far red. And if you're missing your M cones, according to kind of this simplified theory, you should be fine because the other two will allow you to see, your blue and most of the range of like red and these kind of middle wavelengths. What you will be missing is any color that comes from the combination of these two or the combination of these two. So if you miss your M cones, you won't be able to distinguish like green or cyan colors. And then can you predict what colors you'll see if you stimulate different combinations of your cones? So if you stimulated your M and your L cones equally, that will allow you to see like a really light green or yellow. If you stimulate your S cones and your M cones about equally, you'll see that light blue or cyan color. If you stimulate your L cones and your S cones about equally, you'll see a purplish magenta color. So I kind of highlighted that here. This is showing you what you see if you stimulate a combination of each of those cones. Questions on that?
No, thank you. Actually, one thing you said, if you're missing your S cone, then you can still see everything okay, except for the overlap.
I was saying if you miss your M cones, you would still be able to see like throughout the full range of wavelengths, but you wouldn't be able to distinguish colors that rely on you comparing the S to the M or comparing the L to the M. So basically what you would be missing is say like yellow and cyan, right? You would just have blue and one other color over here, which tends to be yellow.
Okay. Gotcha. But you said if you lost your S cone, you'll be okay. You can still see.
Well, if you lost your S cones, which is this one, you would miss blue.
Mm-hmm.
You would
Okay.
miss blue. So you could still see green and yellow and orange and red, but you wouldn't be able to see blue. You also
And
wouldn't.
if you lost your M cone, you wouldn't be able to see green, or you could still see green?
You would not be able to see green. You would not be able to see yeah, green because that requires the comparison between the two. And you would not be able to see dark, deep red, and you would not be able to see purple or magenta.
If you just lost your M cone?
Yeah, because that purple magenta is a combination of S and M. So if you're missing,
From the overlap?
sorry, not, if purple is S and L, sorry. Yeah, you need both of them.
Okay. Okay. And you know that from looking at the graph because S goes over purple, but L doesn't go over to purple. So how do you determine that?
Yeah, that's just something that in this class, there is more known about why that is, which I can talk about later, but it's a little bit complicated and out of scope for the course.
Okay. So as far as like question 2, this would be perfectly reasonable to say if we lost our L cone, then we can't see red or orange. If we lost M, we wouldn't be able to see green. And if we lost S, we can't see like blue, purple. Is that reasonable?
Yes.
Okay, thank you.
Yeah, Kaylin.
Is the reason that for like you wouldn't really lose much if from the M cone, is that because the M and the whatever the red cone is because there's so much overlap in the wavelengths between those, like the wavelengths they cover?
Yeah. And to be clear, when I mean you're not losing a lot, I meant like you would be able to respond to almost the same range of wavelengths. But in terms of color, you would lose quite a bit because instead of seeing all of these different colors, you would basically have 2 different colors you could see.
Okay, thank you.
You're welcome. All right. Last thing about vision is just be aware that other species can see, or not see, but they have photoreceptors that are sensitive to electromagnetic radiation that humans cannot see. So that is totally possible that other animals can see like UV light. And of course, by see, I don't necessarily mean see in the sense that humans see, just that they can respond to it. All right, and then the last topic for sensation is plant defenses. So you need to know, how do plants sense and respond to herbivory? And also, given that pathway, if there's some kind of change to the pathway, can you predict other changes that might happen? So the pathway you need to know is in response to some kind of herbivory. So something is damaging the plant cells. That's going to activate receptors on the plant. So sometimes it's like saliva from the caterpillar. Sometimes when the plant breaks, the plant cells will like spew its guts and amino acids come out, and that's what binds the receptor. It doesn't really matter. Something about the damage is going to release some signal that binds to a receptor. That's going to cause a signal cascade that will release jasmonate, or JA, a hormone. And that hormone will travel within the cell and also out all across the plant. And if there's receptors for it, then JA will bind to the receptor. And that receptor is a transcription repressor. So in the diagram, the little tiny circle is JA, the hormone. The bigger blue circle is the repressor, and it's also JA's receptor. And when JA binds to that receptor slash repressor, the repressor comes off of a transcription factor, which is the brown thing. Once the repressor comes off, then that transcription factor can bind to genes and increase transcription. Yes, question.
I was just wondering, in how much like depth do we need to know this process? Because I know like it's kind of simplified when it comes to the sense of jasmonate causing the repressor to unbind. Like in the book, it says like binding of jasmonate to receptor leads to degradation of the Jaz receptor. Like, do we need to know it in that much depth, or do we just need to know that like jasmonate binds and allows for the transcription factor to bind to the promoter and cause transcription to occur?
Exactly that. I would know that in general, herbivory causes JA hormone to increase, and JA is going to cause the transcription factor to be able to bind and increase transcription. So herbivory is going to increase transcription. And then if you're given the pathway, like say, like you have the diagram for the pathway, and I say something's going to change, can you tell me the result of that change? So for example, what would happen to transcription if you block the JA receptor binding site? So if this little dot is JA, and when it binds, it removes the repressor. If it can't bind, then the repressor won't be removed. Repressor stays on, transcription will not increase. Another example would be, what would happen to transcription if you increased the total amount of the JA hormone? If you increase the total amount, then it would bind more receptors, less repression, more transcription factor binding, and more transcription. So again, you should just be able to reason through that given the diagram. I'm not going to ask you, tell me the steps of this pathway, but given the pathway, can you predict changes? And that is the end. So any questions?
Is there no time stuff on the exam, like the plant timing stuff?
No need to worry about plant timing.
That's awesome.
Anthony, yeah.
I was wondering if the repressor just doesn't work in general, then the addition of jasmonate just wouldn't affect anything, right? Because then transcription would just be constantly occurring.
Say that again, sorry. What was it?
If there's like, if the repressor, like let's say the repressor is broken, like something happens to the repressor, then there's nothing repressing the transcription factor from binding to the promoter, right? So that the amount of jasmonate just wouldn't be related, like it wouldn't matter because jasmonate's not binding to anything.
Yeah, if there's a mutation to the repressor, then I would predict the transcription factor would be binding and there would be lots of transcription, regardless of whether there was herbivory or JA or anything. Yeah, question?
I was wondering, can you go over again the graphs for honeybee and humans?
Yeah. So my point here was just to show that bees have photoreceptors that are responsive to electromagnetic radiation that is different than humans, right? So they can see over a wider range than humans can. So they can actually see into the UV range, which humans cannot. And so basically just be aware that different animals have different receptors and what wavelengths they can see depend on those receptors. And of course, see does not mean perceive in the way humans perceive, right? You should really not say see, you should say responsive to, because see kind of implies perception. Yeah, question?
Oh, I was wondering, do we need to know about PFR and all that? Like,
No, that will come up on exam 4 for sure, but not for exam 3.
could we go over the Jasmine again?
Yeah.
That whole process. You said that we need to determine what happens if something doesn't work.
Yeah. So it's helpful to know what the pathway is in order to do that. So basically, when the plant receives some damage from an herbivore, that's going to cause some response. Say the saliva binds to a receptor on the plant. That receptor causes a signal cascade that results in synthesis of JA. JA can travel and within the same cell or distant cells, because it's a hormone, it will go over and bind a receptor, that is the little blue thing. And when it binds, it removes that receptor from this brown thing. So the blue receptor is a repressor of this brown thing, which is a transcription factor. So normally this repressor is binding to the transcription factor. So the transcription factor can't go and increase transcription. But when JA binds, the repressor is removed from the transcription factor. So JA removes the repression of the transcription factor. So then the transcription factor can bind to the gene and increase transcription.
Okay, gotcha. So you said the blue one is the repressor, and then
Yeah.
JA binds to the repressor, so it goes off
Yes, yeah.
the. Okay, gotcha. And the brown thing, you said that's the transcription factor?
Yeah.
Okay, gotcha. So that increases transcription of whatever defense mechanism.
Yeah, exactly.
Okay, thank you.
You're welcome.
Quick question from the like the HAMP down to the part where it enters the cell. Is that just like a channel, or is it like is it gated? Is it just like a free flowing channel, or is there some like ligand gated, or like
Like
a
here to
ion, like
here?
something? No, after that, the step where it enters the cell, like
Oh
the
yeah.
lumen, the purple, where it goes from orange to purple, that spot.
Like here?
Slightly lower.
Here.
Yeah, that part. Yeah. Is that
Oh,
channel just like open, or is that a channel?
it's a like kind of a hole in the plant membrane called plasmodesmata, and it allows just plant cells to exchange things between their cytoplasms. So if one cell makes a bunch of hormone, some of it will leak out and move into other plant cells.
So
So
this is the one that was about the electrical signals, right? Like electrical signals travel through the plasmata as well,
electrical
right?
signals do too, yeah. So electrical signals do, hormones do, yeah. And hormones can also travel even further by going into the phloem. So I almost want to say this is
So
to the
can
phloem because it's a different color. I changed my mind.
mhm. Can this be altered in any way? Like can there be mutations here, or are we just like is it
Yeah,
just
I
like
could
a
see,
spot
yeah,
that's
an
all?
example of like, what if um, the channels between cells were blocked? What would that do? That would just keep the signal local.
That makes sense.
Yeah, another question?
You might have already answered this, but do we have to know about plant responses to light for this exam?
No. So you should know different organisms respond differently to light, but I don't think any specific things to plants come up. No.
Okay, thank you.
You're welcome. Yeah,
I
another
think
question?
you already explained, yeah, I think you already explained this, but just to make sure, so if we lost, if we lose our, like, if we lose our L cone, then we can see red and orange.
Yes, yeah, exactly.
And for S is blue and purple,
Yes, yeah.
and for M is green, deep red, purple and magenta.
Yes. So if you lost your M or your middle cones, you wouldn't be able to see green or cyan.
I see. Can we go through like different variation of question 3? Like,
Yeah. So if you stimulated your M and L cones about equally, then you would see yellow. If you stimulated your S and M cones about equally, you would see cyan. And if you stimulated your L and your S cones about equally, you would see magenta. So those are the ones to know. That's what's shown here. So M and L, you see yellow. L and S, you see magenta. S and M, you see cyan. All three of them together, you see white.
okay, that makes sense.
Yeah, question?
So if you stimulated one cone more than another, would the color that you see be different than if they were both stimulated equally?
Yes, exactly. So like if you
How would we?
if you stimulated the L cone more than the M, then instead of seeing yellow, you would probably see something more biased towards the L cone side, so orange. So if
Okay.
you stimulated like S and M, equally cyan, a little bit more S, I would say bluish cyan or greenish cyan are things you might say.
Okay, got it. Thank you.
You're welcome. Question?
I was wondering, can we go over the one example that professor posted on scaling?
On scale? Which one?
I think he posted on Piazza.
Sure. Let me see if I can find it. Um, so where on Piazza did he post a question?
It's post number 202, 202.
Okay. Is it where it says exam 3, one more practice problem?
Yeah.
Okay. Number 3, like most amphibians, salamanders breathe mostly through their skin. About 4 feet long, about 1 inch long. Relative to the Chinese elephant, what is the approximate skin area? Okay, yeah, so this is just kind of the same idea. So he gave you going from 1 inch to 4 feet, or actually it's backwards. But you would find what number in do you need to use to go from 4 feet to 1 inch? So first thing you would do is you would get the feet into inches, right? And then figure out by what number do you need to multiply to go from 4 feet to 1 inch. So how you do that is 12 times 4 divided by 1. So it'd be 1/48th. Although it looks like he's saying 1/40th, which is kind of weird. It should be 1/48th. So in would be 1/48 because that's the number that would allow you to go from 4 feet to 1 inch. And then it says, relative to a Chinese giant salamander, what is the approximate skin area? So does he give you the original area? No. So to get area, you would have to do, because area is a two-dimensional thing, you'd have to do in squared. So you would have to do 1 divided by 48 squared. 1 divided by
Could you do this on a whiteboard or something?
I don't have my pen today, so I can't draw out because my
Okay,
isn't working very well.
okay, no problem.
But it's the same process as like the salmon or the example I gave. Yeah, it's really just the same thing where you find out what in is, and then because it's asking for a two-dimensional value, you're going to do in squared. If it's asking for a three-dimensional value, you do in cubed.
So for part A, we convert the inch into feet first,
I would
and then I'm still confused on how is he getting 140.
I'm not sure where he's getting 1/40th either. I don't know if that's just, it says EG, so I don't know if that's just an example. I think that's
So like, what
it
would
is.
be the right? So what would be the right process to get 10?
So basically you would convert 4 feet to inches. So you do 4 times 12, which is 48, right? And then you want to know 48 times what gives you 1 inch. So that would be 1/48. So I think he's just saying, for as an example, your answer could be 1/40th as much, but the real answer is 1/48 as much.
Let's see. Wait, how did you know to do 1/48 and not 48/1?
Because if you times 48 times 48, you don't get 1. But if you times 48 times 1/48, you do get 1. So I was trying to see, how can I go from 48 inches to 1 inch? And in order to do that, I need to times 48 by 1/48. So right, like 48 times what equals 1, right? So you can do 48x equals 1. And then just do algebra and you get x equals 1/48.
And that would be our end in this case.
Mm-hmm.
Okay, that makes a lot.
So then if that's your in, from there you can calculate area, you can calculate mass or whatever value he gives you. Cool. All right, that is the end of the review. So I hope this was helpful, and I'm happy that I can host this and it won't be 3 hours. So yay. All right.
Thank
Good luck
you.
studying.
Thank you. Thank
There
you.
was, thank you.
guys.
One quick question. There was a thing about Reynolds numbers. Is that going to be on the exam or no?
No,
No? That's great. Awesome. Thank you.
you're welcome.
And I also had a question. Everything about like PFR and like constants and everything is going to be on the next exam, not this one?
Yeah,
Awesome. Thank you.
you're welcome.